Hello everyone, welcome to Banajawad PU online classes. Already in the last class we had discussed about the different kinds of the contraceptive methods and merits and demerits of contraceptive methods and all. Now after that we have discussed even MTP, what is MTP, merits and demerits of the MTP, clearly we discussed in the last session. Now in this session we will start with a new topic or the new concept that we can say it as a amniocentesis. Amniocentesis. What is mean by the amniocentesis? How it is useful for the mankind and what are the drawbacks of this amniocentesis? We'll discuss first. Later on we'll move to the sexual transmitted diseases. Okay, what are sexual transmitted diseases? What are the pathogens are causing these different kinds of the sexual transmitted diseases? What is the mode of transmission? We'll discuss clearly. Okay, right. Now we'll move to the amniocentesis concept. Amniocentesis concept. What is amniocentesis actually? Amniocentesis is a technique. It is a technique. It is a prenatal technique. Prenatal means before delivery we are identifying the baby, whether it is a girl or boy or any kind of defects are present in the baby by using a chromosomal pattern of the baby. Clear? Now we can see it is a prenatal diagnostic technique used to determine what we are going to be determining here. First one, we are determining sex of the fetus and congenital diseases. Congenital diseases means Congenital diseases means which are the diseases are inherited from their parents to baby. Such diseases we are identifying by using this amniocentesis technique. First one, we are identifying sex of the baby. Sex of the baby means sex of the baby in the sense whether the chromosomes, already I told you that it is a prenatal diagnostic technique to determine the sex of the baby. How we are going to be determining the sex of the baby? Means by using the chromosomal pattern. How we are going to be identifying is, for example, the chromosomal pair is XX chromosomal pair is there, we can consider it as a female baby. And if it is the chromosomal pair is X and Y, we can consider it as, or we can say it as a male. Okay, clear. Now, congenital disorders or congenital diseases. Congenital diseases means the diseases which are passed from their parents to their children. Such diseases we can call it as a congenital diseases. What are the examples for this congenital diseases is, congenital diseases we are discussing here, that is the Down syndrome, Turner syndrome and Klinefelter syndrome. These are the very important examples for the congenital disorders which are mostly acquiring from their parents to the babies. About this clearly we will discuss in the next chapter, principles of inheritance. Just here we are explaining only the examples. Clear? Now the next one, we are also determining by using this technique metabolic disorders. Metabolic disorders means here the metabolic disorders which are getting to the baby is, one is phenylketonuria, second one is alkaptonuria. Phenylketonuria and alkaptonuria, these are the disorders acquiring to the baby due to the lack of phenylalanine hydroxylase enzyme and due to the lack of homogastric dehydrogenase enzyme. Because of this lack of these two enzymes, the baby will get the two disorders, that is the metabolic disorders. One is phenylketonuria, second one is alkaptonuria. Clear? Now, to identify these three means one is sex of the fetus, second one is congenital diseases, third one is metabolic disorders. We are using the special technique called as a amniocentesis technique. Clear? Now, how we are going to be identifying, how we are doing this test, we will discuss now. That is, we can say the procedure of the amniocentesis. Procedure of the amniocentesis. In this, I divided stepwise to understand the students. So I divided into stepwise. I will explain now. We can see here. Step 1. First of all, we have to identify. We need to identify where the fetus is presented in the mother womb. Where the fetus exactly located. To identify the location of fetus, identify the location of fetus is the first thing. That is the step 1. How we are going to be identifying the location of fetus? By using which one? That is by using ultrasound or by using sonography. By using sonography or by using ultrasound waves, we can identify where the fetus exactly located. After identifying the location of fetus, what we are going to be doing is, we are injecting a small narrow needle into the amniotic cavity of the mother and amniotic cavity of the fetus and collect the amniotic fluid. Collect the amniotic fluid which contains fetal cells and as well as enzymes. Are you getting now? First step what we are doing is here identifying the location of fetus. In the second step what we are doing is here we are collecting the amniotic fluid which contains fetal cells and enzymes. How we are going to be collecting? With the help of a hollow needle. With the help of the hollow needle, we are going to be collecting this amniotic fluid from the amniotic cavity of the fetus. Clear? Now, the third step what we are doing is, after collecting amniotic fluid, we have to culture it. We need to culture it this. In vitro culture. In vitro means nothing but outside the body. We are going to be culturing in the laboratory. After culturing, what we are going to be doing is, we are doing the final step, that is the karyotyping. You know, karyotyping means nothing but karyo meaning is nucleus. Typing means nothing but here. What we are going to be doing is, in this area means, mainly we are going to be taking the pictorial pictures of chromosomes. For example, one pair of chromosomes here, you can see that. Pair of chromosomes, first pair, second pair of chromosomes, pair 1, pair 2, like that. All the 23 pairs of chromosomes we are going to be taking in the form of a picture. Then finally, we are identifying whether any kind of defects are present in these chromosomes, whether it is more number of chromosomes are there in one pair or less number of chromosomes are there in one pair. We are going to be identifying by using the karyotyping technique finally. Are you getting now? One more time I will repeat it now. See that. Step 1, we are doing identification of the fetus. Identification of the location of fetus. Step 2, we are collecting the amniotic fluid by using hollow needle. And step 3, culturing of in vitro, culturing of the fluid which we are collected from the amniotic cavity. Now, finally we did the step 4. Step 4, that is we are doing here karyotyping. Karyotyping means nothing but the chromosomal pattern is we are taking the pictures, and by using the pictures we are detecting under the microscope, we are observing whether it is any kind of the defects or disorders or kind of we can see with this by using this one we are identifying these 3 sets of the fetus by using chromosomal pattern, and congenital diseases we are identifying by using chromosomal pattern. Metabolic disorders we are identifying by using chromosomal pattern. 23 pairs of chromosomes are perfectly presented or not. We are going to be identifying whether it is not presented properly, what kind of the defect is present in the baby, we are identifying it here. Clear? This is the procedure of amniocentesis. Okay, now we'll move to the importance of amniocentesis. What is the importance of this amniocentesis? We'll discuss now. Importance of amniocentesis is, we know sex determination. First one is the sex determination. By using this technique, we are identifying the sex of the baby, whether it is male or female. If the chromosomal pair is X and X, we can consider it as a female. If the chromosomal pair is X and Y is presented, we can say it as a male. We can say it as a male. Now we can see, this is with the help of this one, we are identifying the sex of the baby. By using chromosomal pattern, whether it is X and X, we can call it as a female. X and Y, we can call it as a male. Now, second one, we are determining the congenital diseases. Determining the congenital diseases. Already I told you that congenital diseases means the diseases which are passing from their parents to babies through the inheritance process. That is, examples of some of the congenital diseases I have given here. That is, first one, trisomy condition. What is meant by the trisomy condition? For example, if you take here 21st pair of chromosomes. 21st pair of the chromosomes. Two chromosomes are present. Pair means nothing but two chromosomes. There, if you observe one X chromosome and as well as one more X chromosome presented in that area, and here what happens is increases the one more chromosome in that area. If the three chromosomes are there in that one place, we can consider it as a trisomy condition. In the trisomy condition time, what we are going to be observing is, what we are getting is mainly Down syndrome disease. This Down syndrome disease we are getting because of the trisomy, because of at the 21st pair of chromosomes is having one extra chromosome, whether it is X or Y, one extra chromosome is presented here. Because of this extra chromosome, the baby will get the Down syndrome disease. Okay, clear. Now the next one, Klinefelter syndrome. Klinefelter syndrome means nothing but feminine characters. Male is behaving like as a female, female is behaving like as a male. Here especially we are observing in the males these characters. Klinefelter syndrome we are seen in males. The male is behaving like as a female. What happens is here two X chromosomes are presented and one Y chromosome is there in one pair, then we will get the Klinefelter syndrome. If we are observing in the karyotyping time, these pattern of chromosomes if we are seen, the person definitely having a Klinefelter syndrome. And lastly, Turner syndrome. Turner syndrome, it is especially for the females. We are seeing here, Turner syndrome means the person is unable to conceive. The person is unable to conceive. The person is not having an ability to get the pregnancy because of Turner syndrome. Here, Turner syndrome means lack of one X chromosome. Here what happens? Extra one X chromosome is there in the male. Here X and Y is male we can say, but here two X chromosomes are there. That's what the male is behaving like as a female. But in case of Down syndrome, one extra chromosome is presented. In the pair, two chromosomes are there, but in this case, one extra X chromosome is there, that is causing the Down syndrome process. And finally, the Turner syndrome. In the case of Turner syndrome, one X chromosome is less here. In the female, two X chromosomes are there, but here, absence of X chromosome, it leads to a Turner syndrome. Now the final one, determination of metabolic disorders. Here, what kind of metabolic disorders baby will get oftenly means, first one is phenylketonuria. This disorder the baby will get because lack of phenylalanine hydroxylase enzyme. Due to the lack of this enzyme, the baby will get the phenylketonuria metabolic disorder. Due to this, accumulation of the phenylalanine takes place in the baby, sometimes cause the brain damage, as well as sometimes the baby will get the seizures. Seizures means nothing but psychological disorders. Okay, now finally one more metabolic disorder is alkaptonuria. Alkaptonuria is also one of the disorders. In this case, how it is alkaptonuria causes to the baby is because lack of homogenistic dioxygenase enzyme. Due to lack of this enzyme, the baby will get the phenylketonuria metabolic disorder. Clear? Because of these all the things, because all the disorders we will discuss in the next chapter clearly with the symptoms, preventing measures and all the things. But here just we are discussing only the examples which are belonging to the congenital diseases, examples which are belonging to the metabolic disorders. Okay, clear. Now, the lastly we will discuss about the drawbacks. Drawbacks of the amniocentesis. What is the importance we discussed and what are the drawbacks are there for this amniocentesis we will discuss now. Drawbacks of amniocentesis is misused to determine the sex of the fetus. You told to identify the sex, no sir. Again you are saying it is a desired drawback, because the reason is if the person identified fetus is female and then she is aborted. I told you that illegal female feticide mainly causing due to the, by using the amniocentesis we are identifying the female fetus. After that we are going to be killing killing mostly the female fetus. That is also one of the drawback because of this amniocentesis. Amniocentesis drawback is amniocentesis is misused. Some members, not all the peoples, some members are misused to determine the sex of the fetus, so that amniocentesis is banned in India. Amniocentesis is banned in the India because the reason is illegally using this amniocentesis in the India, especially, so that the reason the amniocentesis are banned in the India. These amniocentesis are banned in India in the year of 1994. These amniocentesis are banned in the India. This is also asking for the 1 mark question, and sometimes they are giving in the entrance exams. Clear? This is about the amniocentesis story. Okay, are you getting now? Clear. Right. Sexually transmitted diseases. Here, sexually transmitted diseases, in the name itself they indicated sexually transmitted. These diseases which will pass through the sexual contact. Now we can see the perfect definition of the sexual transmitted diseases. The diseases are infections. Which are transmitted from infected person to normal person. It is not coming from the normal person to normal person. It is passing through the infected person to normal person through a sexual intercourse, sexual contact. Sexual intercourse in the sense sexual contact or sexual participation. Okay, are collectively called as STDs. Or else we can say it as a venereal diseases. What is the meaning of venereal diseases mean? The diseases which will occurs to the genital organs. The diseases which will cause through the genital organs, that we can say it as a venereal diseases. The diseases which are caused to the genital organs, those diseases we can call it as the diseases which will occurs to the or which are causes to the genital organs. Such diseases we can call it as a venereal diseases, that we can simply say it as VD. Now ORR, these diseases are also called as a reproductive tract infections. That is, we can say it as a RTI. RTI. Sometimes these names also, by using these RTI also they may ask the definition, they may ask the question. By using the VD also they may ask the question, or similarly they may ask the question in the name of STDs. Clear? Okay, we have to know other name of STDs also. Okay, right. Now we can see how these STDs are causing, what are the common STDs we are observing in the, mainly in the person, we are observing what are the common diseases we are seeing now. Now we'll discuss in our chapter whatever the diseases they given, that only I have written here, because some more extra diseases are there which are caused due to the even protozoas. Okay, but I did not mention those because as per our NCERT syllabus which they mentioned, I am explaining those only. Now we can see name of the disease and pathogen which is causing this disease. Now we can see first one we are explaining about the bacterial diseases. Bacterial diseases. Bacterial diseases, first one, syphilis. Syphilis is a bacterial sexually transmitted disease. It is causing due to the Treponema pallidum. Treponema pallidum. Treponema pallidum is a organism, microorganism, that is especially bacteria which will cause the syphilis disease. Syphilis disease. Now symptoms we will discuss commonly afterwards. Now we can see next disease is gonorrhea. Next disease is gonorrhea. The gonorrhea is caused by the Neisseria gonorrhea. Neisseria gonorrhea is a microorganism, that is bacteria which will cause the gonorrhea disease. Now the next one, chlamydiasis. Chlamydiasis disease which is caused due to the Chlamydia trachomatis. Chlamydia trachomatis. Chlamydia trachomatis, which is a bacteria which will cause the chlamydiasis disease. Chlamydiasis disease. Okay, fine. Right. The next one, viral diseases. Viral, the virus which is causing the sexual transmitted, those are the names and causative organisms we'll discuss now. That is, first one, herpes genitalis. Herpes genitalis, it is a sexual transmitted disease which is caused by the HSV. HSV means herpes simplex virus. Herpes simplex. Herpes simplex virus which will cause the disease, that is herpes genitalis. Okay, it is especially DNA virus. DNA is a genetic material in this virus. Viruses are two categories, RNA viruses, DNA viruses. Mostly we have seen RNA viruses, but some rare cases we are observing the DNA viruses. The DNA viruses example is herpes simplex virus. Sometimes they will ask the even in entrance also. Herpes simplex virus is a DNA virus or RNA virus. Okay, right. The next one, AIDS. This is well-known disease to all the peoples. AIDS is caused by the HIV virus. These HIV viruses belong to the family of retrovirus category. Retrovirus category. Now the next one we will discuss hepatitis B. Hepatitis B, it is causing to the liver, especially it is affecting on the liver. It is also caused to the sexually transmitted. It is also possibilities to get through the sexual contact. That is hepatitis B caused by the hepatitis B virus. Hepatitis B virus. Now lastly, genital warts. Genital warts. These disease caused by the human papilloma virus. Human papilloma virus, that is, we can say it as a HPV. These human papilloma virus is causing genital warts, as well as these virus we have seen in case of the cancer. In case of the cancer also we are observing this one, that we are explaining in the human health and diseases. Okay, oncogenes are causing the cancer. That oncogene conversion is caused due to the mainly papilloma virus. Clear? Now this is the disease names and causative organisms which are causing different type of the diseases the body. And according to NCERT, they mentioned only the name of the diseases, but I given even pathogen names because in the entrance exam they are asking pathogen names also even. Clear? Now next one, mode of transmission. How these sexual transmitted diseases are passing through the person to person and what are the ways are there for that. Now we can see those one by one. First one, STDs are generally transmitted through sexual intercourse. These diseases are generally passing through the or passed by the sexual intercourse. But in some cases, some other ways also it may cause or it may transfer to the one person to another person. What are those other ways is another one, that is using of infected persons used needles. For example, one syringe is infected person is used. The same syringe is another normal person if they use, that person also may get the chance of getting the sexual transmitted diseases. That is the disease which are given the list. No, those diseases they may get the chance is there to get. Now surgical instruments in the sense whatever the instruments are using to do the surgery. For example, doctors are using some instruments to do the surgery for the infected person. The same instruments without proper sterilization, without proper cleaning, if they use to normal person, chance to get these diseases. Chance is there to get these diseases. Clear? Now the next point, by blood transfusion. By blood transfusion. This you know very well. For example, if we have normal fever, if we go to the hospital, okay, doctor will test the, first they will take the blood test. In the blood test, they will test even malaria, HIV positive or negative, TB, everything they will test it. The reason is, if the person may have the AIDS disease, that blood if you give to other person, they also get the AIDS disease. That is the reason we need to check out properly this blood before giving it to the other person. That we will discuss in the preventing measures. Now by using the blood transmission also the person may get the HIV and these diseases. Now the last one, that is through the infected mother to fetus. This is also possible because while rupturing of the fetus, they may the rupturing of the fetus, mixing of the mother and fetus blood takes place. At that time, chance is there to get the disease from the mother to fetus. Chance is there to pass the disease from mother to fetus. This is a ways to pass the diseases. These are the diseases are passing ways. These are one more time I will repeat it. Mode of transmission of these diseases is by using, first one is the STDs, sexual transmitted, means generally transmitted by the sexual intercourse. Second one is using of infected persons used needles or surgical instruments. And third one is by blood transfusion. Fourth one is through infected mother to fetus. Okay. We are discussing mode of transmission, how the sexual transmitted diseases are passing from one person to another person. We discussed already. Here few more causes transmission also we can say, that is due to unprotected sexual contact, one of the mode of transmission. That is also one of the mode of transmission we discussed. And after this we will move to the symptoms. What are the symptoms of these sexually transmitted diseases? Common symptoms we are discussing, especially we are not going to be discussing 1-1 disease and symptoms of the 1-1 disease we are not discussing. Commonly what are the symptoms we are getting because of these diseases we will discuss now. That is, itching is one of the common symptom. Second one is fluid discharge. Fluid discharge means if the genital organs are getting damaged or infections, leak means fluid discharge, like pus releasing and blood means bad. Sometimes what happened is fluid is going to be discharging, that is because of the itching, because of the infection causing in the reproductive organs or genital organs we can say. and that is in the form of a pus. Now the third one, if you see, swelling in genital region. Swelling in genital region, because I told you that if in case any bacteria or virus attacking into the genital organs or damaging to the genital organs, there we are noticing first one, swelling. Swelling takes place. And the next one, fourth, slight pain during urination. Because of the damage, because of the swelling, because of the fluid discharge, what happens is here, finally slight pain will occur during urination time. Are you getting now? One more time I will repeat it. Symptoms which are causing due to the sexually transmitted diseases. The symptoms are, first one is itching is one symptom. Second one is fluid discharge. Third one is swelling in genital organs or genital regions. And fourth one is slight pain during urination process. Clear? These are the symptoms which are caused due to the sexual transmitted diseases. And if the symptoms are detected early, we can treat the sexual transmitted disease even. But if in case these symptoms are not detected early, what are the complications occurring into the person, occurring into the peoples, we'll discuss now. Some complications of STDs. If you are not detecting early, what kind of complications STDs will cause to the person, infected persons, that we'll discuss. First complication is pelvic inflammatory diseases. Pelvic inflammatory diseases. It is a infectious or inflammatory disease of the upper female genital tract. Upper female genital tract. It includes uterus, fallopian tube and ovaries. Fallopian tube, ovaries. It means pelvic region getting the diseases. The pelvic regions are getting the diseases. That is because of late detection of the STD. That is first one is the that is causing the complication. That complications, first one is pelvic inflammatory disease. In the pelvic inflammatory disease, mainly uterus may be affected, fallopian tube regions are going to be affected, and ovaries are going to be affected. That we can say it as a pelvic inflammatory diseases or disorders, we can say. Now, second complication is stillbirth. Stillbirth means, stillbirth is the birth of a baby who has died. Already the baby died in the mother womb. Such babies where the mother is giving birth, such birth we can call it as a stillbirth. Stillbirth. Okay. Now the next one, abortions. We know because of STDs sometimes may cause the even abortions. And lastly, infertility problem. Infertility problem means the couples are unable to conceive even after participation of 2 years of unprotected sexual contact. Then also the couples are not having an ability to get the pregnancy. The female is not having an ability to get the pregnancy. Such condition we can define it as a infertility condition. One more time I will repeat it. If the female is not getting pregnancy even after participation of unprotected sexual contact up to 2 years. And up to 2 years. Such condition we can define it as a, or say it as a infertility problem. Okay, now the next one. People belonging to age group mainly 15 to 24 years are highly exposed to STDs. The reason I told you problems related to the reproductive health is, this is also major problem because lack of awareness or lack of sex education in the schools, it caused the mainly this problem, that is you can say STDs. Now prevention, how we are going to be treating, how we are going to be preventing these STDs diseases is, first one is avoid sexual contact with unknown partners, with unknown persons or multiple partners multiple partners. This is the one preventing measure. Okay, that is avoiding sexual contact with unknown persons or multiple persons is one of the preventing measure for the sexual transmitted disease. The second one, always use condoms during in coitus or during in sexual contact. Always using these condoms is good for the health, good for the to get rid from the sexually transmitted diseases. Already advantages of condoms term we told this thing also. Condoms are having an ability to prevent the sexually transmitted diseases or else RTI infections. Now third one, in case if you are getting doubt, if you are participating for example by mistake or we want to participate in sexual contact, if you are participating sexual contact with unknown person or multiple person, you are getting doubt in your mind, maybe I am getting HIV or not, like fear you are getting, immediately go to a qualified doctor for early detection and get complete treatment. If diagnosed with a disease. What is the meaning of this one is, if we are getting doubt, if you participate in sexual contact with a unknown partner or multiple partner, at that time if the person is getting doubt, if you are getting a small doubt, then immediately consult a qualified doctor and to detect the disease which we are getting, and immediately you will get the proper treatment for that disease. Okay, this is about the complete history of sexually transmitted diseases.